So it's my great pleasure to now introduce Dr. Dan Tardiff from CAMP4.
All right, thank you, Yuri. I can tell I'm already going to have some formatting problems, so we'll see what lies ahead here. But it's great to be here. Thanks, Mike and Catherine, for the introduction. You know, a number of us were here about a year ago, and you know, we were really interested in Syngap, but it hasn't really gotten to the point that it is today. A lot has happened over the last year for us, and you know, we're really excited about what's coming up. So I'm going to walk you through, tell you a little bit about our biology, because I think we're a bit new to the game here in terms of, you know, there's been a lot of discussion about splice switching oligos, and we're doing something, you know, quite a bit different. So I think it's important that you understand the underlying biology, and then I'll tell you about some of the preclinical work that we have of our development candidate that we're moving towards the clinic. So some forward-looking statements and disclosures. You know, so all of our colors are messed up. Okay, so this is the challenge, right? Haploinsufficiency, you know, we're kind of running on a half-full tank here due to all these nonsense mutations, frame shifts, essentially a 50% reduction in protein level. And what we're trying to do is restore protein, the endogenous protein, and upregulating gene expression by targeting non-coding regulatory RNAs. These are RNAs that are endogenous, they're expressed by all cells, and I'm going to spend some time telling you about what these are so you can understand what we're going after. And our goal is to really increase Syngap protein, increase endogenous Syngap protein to hopefully improve disease symptoms. All right, so now I'm going to tell you a little bit about regulatory RNAs. These are non-coding RNAs that are transcribed from all actively transcribed genes. So they're generated from both promoters and enhancers of all cells. So while an mRNA is being transcribed in the sense direction, in the antisense direction from that promoter, you're actually getting expression as well. And also enhancers, which are more distal genetic elements that actually come in proximity to promoters to help regulate gene expression. These two are actually all transcribed in a bidirectional manner. So in addition to the mRNA, there's a whole bunch of other RNAs that are being generated. And what they do is they actually act locally, we believe co-transcriptionally, and they interact with transcription factors and keep high local concentrations of transcription factors and help regulate gene expression. And they do so within what we call a physiological range, so sort of up and down a couple fold. They're not responsible for turning off a gene or inducing a gene several fold. They're sort of more subtly modulating transcription. Think of them like a rheostat. And they're very specific. So these regulatory RNAs, they're not sort of leaving the side of transcription and going and having an effect on many genes. They're actually very specific for the gene close to where they're being expressed. So what we are really focusing on is, and have kind of worked out, is that we can target these regulatory RNAs with antisense oligonucleotides to increase gene expression. And what we believe is happening is that by targeting the regulatory RNA sequence, we're altering the structure of the regulatory RNA and having some impact on the interaction of these regulatory RNAs with proteins and transcription factors, potentially displacing negative factors or promoting the interaction with positive acting transcription factors. And again, the effect size that we see is, you know, what we would say is modest, but that's exactly what you need within a haploinsufficiency. And I think it's actually important, this idea of getting too much expression, that's not a problem for us. Similar to what, you know, Megan saw in terms of getting maximizing around twofold, that's kind of what we see as well with this mechanism. And I think it's important to point out that when we target these regulatory RNAs, it's highly specific. So we're not having effects on many, you know, whole swath of RNAs, but it's very specific to the gene from which these regulatory RNAs are being expressed. And again, all actively transcribed genes have them. So now I'm going to tell you a little bit about how we identify these, because this is really a key part in the first step in our platform. And this really is what we call refer to as mapping. So we use a whole suite of genomic approaches, looking at histone modifications, looking at chromatin accessibility, nascent and total RNA, as well as three-dimensional architecture of chromatin, looking at how these enhancers that are several kb away come into contact with promoters. And then we can sort of map that and look and use a machine learning algorithm that we've developed to help select which enhancers or promoter elements are specific to that gene. So here, for example, this is looking at a gene, OTC, it's a liver gene for some other work we were doing, and you can see each of these peaks is sort of different enhancer regions that are expressing these regulatory RNAs, but what we're able to do is hone in on which ones are the most specific based on their score. And then if we take a zoom in a little bit more at this one particular enhancer that is boxed in green, once we predict these regulatory sequences, we then actually isolate the regulatory RNAs that are being expressed. So in this case, this is an enhancer. We use probes that are antisense, that are complementary to the regulatory RNA. We pull them down, and then we do long-read sequencing, and then actually can identify the regulatory RNA species. And what you can see in the orange and yellow or green here is that you get bidirectional transcription from this enhancer, and these are the regulatory RNAs that we're going after. All right, so what about Syngap? So we did this for Syngap. We looked at a whole number of different cell types in both cell lines, iPS-derived neurons, and tissue to map the regulatory regions for Syngap. Shown in the upper left are a number of different regions we're focusing in on, and then orange is the regulatory RNA and regulatory region that we selected to follow up on, and validated this across a whole number of different cell types and tissues. On the right, what we're looking at in the first couple lines here, or tracks, are chromatin accessibility in the ATAC-seq. The pro-seq is actually nascent RNA, but in red is the affinity selection of these regulatory RNAs, and then the long-read sequencing, and then sort of stacking all those reads. So we get thousands of reads, we stack them up, and then you can actually see that we get these very well-defined regulatory RNA sequences from glutamatergic neurons, from SKNAS cells, which is a immortalized cell line that we use for screening, as well as human brain. So these RNAs they're in cells in addition, they're in a human brain. About 90% or so are non-polyadenylated, and then about 10% or so are polyadenylated. So once we had this regulatory RNA that we wanted to go after, we screened through hundreds of oligos, did a bunch of SAR, ultimately showing you here the data for CMP002, which is our development candidate that we're advancing. You can see a nice concentration-dependent increase in Syngap mRNA in these cells. Upwards of about twofold or so. This is very robust and has worked across a number of different cell types. And then we can look in patient-derived neurons as well. So what we want to do is look in the haploinsufficient setting where there's a 50% reduction in RNA, as you can see, and then see how much of an increase can we achieve with our oligo. And you can see a nice concentration-dependent increase, getting just about to just north of wild-type levels. Again, I mentioned we are typically around twofold. It's sort of plus or minus there in vitro. In vivo, we're never going above the twofold effect that I'm showing here. So this is nice, showing that the regulatory RNA function itself and the impact of the ASO is conserved from immortalized cell lines into patient iPS-derived neurons. And I think to Ben's point earlier, this is really important, I'm not going to show this, but it is to take that next step and look at functional effects in these patient-derived neurons. All right, so I'm saying that we're increasing transcription, but all I've showed you is steady-state RNA. So are we actually increasing transcription directly? So I'm going to show you a few pieces of data here. On the left here, we're looking at RNA polymerase II by chromatin immunoprecipitation. So this is polymerase II occupancy at the promoter is actually increasing, which you would expect if you're increasing transcription. So that's a nice check that we're increasing the transcriptional machinery. We also use an approach to look at nascent RNA, to label newly made RNA. So in the teal color, this is just the steady state increase in Syngap mRNA with the oligo, and then in orange is basically this nascent RNA you basically label with a modified nucleotide that you can then affinity select through click chemistry and biotin streptavidin interaction. And we're actually increasing nascent RNA directly. And finally, we can look at the regulatory RNA itself. All right, so we're targeting the regulatory RNA, but we're increasing the mRNA, but we also are increasing the transcription of the target itself. So the regulatory RNA that we're targeting is actually increasing as well. And again, we see an increase in the steady state, as well as an increase in the nascent regulatory RNA. And this makes sense since they're sort of all so close together and in proximity. If you're increasing transcription of one thing, you're going to increase of all. All right, so we then want to start understanding what's the impact of targeting the regulatory RNA on the structure of the reg RNA, the interaction with proteins, what's actually sort of at play here. So to start looking at this, we use an approach called shape map, where we take in vitro transcribed RNA, we fold it, we treat it with a nucleotide adduct that will label the different bases based on the accessibility. So is it a paired sequence or is it unpaired? And then we can use sequencing to actually look at where it's modified. We do this in two different conditions, either without ASO or with ASO, and then you can get a map of essentially are there changes in the RNA structure. And you can see sort of in the structure itself, the ASO is binding to the sort of hairpin along with this open loop. In dark blue is very accessible nucleotide sequence, and then essentially white would be fully paired. And on the bottom, what you can see is that there's changes in accessibility sort of immediately upstream and downstream of where the oligo is binding. Essentially what's happening is, the oligo is binding to the reg RNA at this hairpin sort of structure and dissolving it. And really the next question that we would like to answer is what's next, right? So we're changing the RNA structure. Our model is that this change in RNA structure will influence the interaction with proteins, and ultimately it's those proteins that are having an effect on transcription itself. So that's sort of the next bit of information that we're working towards. All right, so now we're gonna go in vivo here. This is data that we shared earlier this year, and we're very excited about generating. So thanks to Ben Prosser for generating this line and sharing it with us. We use his humanized Syngap mouse. Again, this is fully replacing the mouse gene with the humanized gene. This, importantly for us, includes enough of the regulatory region upstream of the transcriptional start site to include our target. And that's a key thing when we're generating a humanized mouse for our target where it's in the regulatory region and not in the gene itself. So we then took the mouse that we got from Ben and then crossed it to heterozygous line, made mice that either have 2 copies of human Syngap or a single copy. And you can see, as expected, that there's a 50% reduction, 50 to 60% reduction in protein level in the heterozygous state. And then when we administer a single dose of ASO by ICV injection, and these are P21 mice, so they're essentially juveniles, we get about a 2-fold increase in protein levels, restoring it back towards wild type levels. And we also get similar results in neonatal mice as well. So this nicely shows that we can target that the regulatory RNA itself and the ASO is having a conserved effect from in vitro into the intact animal. So what we did next was to then explore whether the effect on Syngap levels that we're observing has a functional benefit. So we ran a study in which we administered in neonatal mice with CMP-002 by ICV injection, and we performed 3 studies, 3 different assessments looking at rotarod, wide maze, and a palm reader, which is essentially an open field assay. So looking at motor function, looking at learning and memory, and then looking at the hyperactivity phenotype. So what we saw in each of these instances, one, there was the expected phenotype between the wild type 2-copy human Syngap mice and the heterozygous mice. Again, you can compare the gray to the yellow all the way across. You can see a nice delta there for each of these assessments. And then you can see when we treat with oligo that there's a restoration of these phenotypes back towards wild type. So targeting the regulatory RNA increases Syngap protein and restores function in these assays. The impact on protein you can see on the right. So we're showing cortex and hippocampus. Cortex, we're bringing it right back to wild type levels, a little bit less so in hippocampus. But there's ultimately we're typically in this 1.5 to 2-fold increase in restoring it back towards wild type levels that we see quite repeatedly. So this nicely shows that the impact that we're having on Syngap levels with our oligo is sufficient to increase gene expression back towards wild type levels and mitigate a number of important phenotypes. I will say, just to get out of the head of the question, we weren't able to do EEG on this particular study or seizures, so we just have the motor, we just have these behavioral phenotypes. Right now we're in the process of generating seizure data and EEG data. All right, so then we wanted to explore monkeys, right? So I think there's a few key things. One is the route of administration, right? So in all these mouse studies, we're doing ICV injection. That's not our plan in humans. But IT, intrathecal administration, is been used widely for oligos. There may be some point at which you could do systemic administration with an oligo, but we're not there yet. So we wanted to do IT administration in monkeys, make sure that we're getting sufficient biodistribution, getting sufficient distribution to the regions of the brain that are important for Syngap-related disorders, and have an impact on protein. Now, monkeys are wild type in nature in terms of they have 2 copies of Syngap, so this is not necessarily a disease model, but we can look at distribution, tolerability, and increases in Syngap itself. So This is showing, our target capture data from monkey brain. So you can see that the regulatory RNA itself is very highly conserved. It's positionally conserved. It's 95% sequence conserved. And the oligo is 100% complementary to the monkey sequence. And then in NHP iPS-derived neurons that we generated, we can see a nice concentration-dependent increase in Syngap upon treatment. So we use this oligo to, again, bring this into an NHP study. So we performed a study in which we administered by intrathecal injection, 4 per group. We did 3 different dose levels that I'll just refer to as low, mid, and high. And then we dose every 2 weeks, sort of a bit of a loading dose kind of paradigm, and then looked 2 weeks later. And then we really looked at many different brain regions for both oligonucleotide concentrations as well as increases in Syngap. You can see a nice dose-dependent increase in ASO concentration in multiple different brain regions upon IT administration, very dose linear and predictable and quite nice. There are some other regions, some of the deeper brain regions, you get less ASO in, but those are not as relevant for where we need to target for SRD. So then when we look at Syngap protein levels, you can see a nice dose-dependent increase of about 1.5-fold across frontal cortex and hippocampus. We have comparable data for many other brain regions, but just showing sort of 2 key ones here. And there's some variability, but again, these are wild-type animals in the context of what I think we feel is some kind of downward pressure to keep from overexpressing, but we're seeing a very reliable increase in Syngap, which we're quite excited about. And throughout our development, I think one of the most important things for us is that when we move into the clinic, we're going to have an oligo that's safe, right? That's sort of first and foremost. So, during our entire drug discovery efforts, we're really paying attention to both in vitro and in vivo tolerability. The ASO is very well tolerated in this setting, and we feel quite good about the effort that we've made to make sure we have a high-quality, safe oligo moving forward. All right, so with that, I would like to summarize what I've told you and where we're at. So we've identified this regulatory RNA that is expressed in human cells, in non-human primates. We can administer this to a humanized haploinsufficient mouse and reverse a number of different phenotypes that have been demonstrated by others, and some of which are analogous to what Megan was just talking about as well. We can increase Syngap in non-human primates when we administer by intrathecal injection, and the ASO is very well tolerated. So we initiated GLP toxicity studies in rodents and monkeys in the 3rd quarter of this year, and we are planning a global phase 1/2 clinical study to initiate as early as the second half of next year, pending the outcomes of the GLP tox studies. So, we're extremely excited about this. We really appreciate all the efforts. Oh, what? Okay, my thank you slide is gone. But really, thanks to the entire CAMP4 team and our supporters. Thank you to Cure Syngap. You've been a great advocate along the way for us and helping us think through things. Thank you to a number of folks in this room, sort of researchers and clinicians that have been helping us. I can't remember. And the most important thing is really thanking patients, families, parents, caregivers. I mean, this is why our team comes to work every day and what we're really trying to strive to do, and really appreciate all your support and look forward to things to come. So, thank you.
All right, thanks, Dan. All right, questions? Back there.
Hey there, great talk. R.P. Saunders, right here, from the Volm Institute. Of course, you have 2 copies of Syngap in each cell, and so do you have a sense of, for targeting these regulatory RNAs, the ASOs, whether from a single cell perspective, you're boosting transcription of both the wild type and the variant bearing copy, or are there trade-offs within each cell, and how do you imagine that's going to manifest across many individual cells?
Yeah, I think Ben referenced this as well, is that oligos are not allele selective, right? So we're increasing, we would be increasing expression from both copies of Syngap, so both the wild type as well as the mutant. Obviously, most of the mutations in Syngap are nonsense, and they're not even generating any protein likely. So, but we would be theoretically inducing transcription from both alleles. And I think, to extend that, again, in other cell types, we're not turning on the gene. So when you think of the distribution of the oligo, again, we're not going to be inducing Syngap in genes that are not normally expressing, in cells that are not normally expressing Syngap.
It looked like the regulatory regions were highly conserved across species, but are there polymorphisms in human genomes that could lend themselves to more selective allele-specific ASO targeting?
If you're lucky, theoretically that's possible. I think, that's when you're trying to do allele selective editing or anything like that, you look for those. We do look at SNPs within our target sequence during our development process. There's nothing obvious that we're able to go after for allele selective here, but that's definitely, it's a good question. And maybe I'm just going to get out ahead of any other questions on isoforms, is that with our approach, we're actually, we are increasing isoforms, all isoforms sort of equally to the best of our knowledge. So we're not, having a selective effect on different isoforms from what we understand.
Hello? Yeah, this is Shin Tang from Boston Children's. So it's a terrific talk. Thanks for sharing. I have a question. I noticed that for both your mouse and NHP study, this cortex, like there's a more effective size in terms of this gene expression elevation compared to hippocampus. So could you speculate is there a cell type composition? Is it like this baseline level? So what's different between this brain region?
Yeah, so, if you look at Syngap levels themselves from what we've done and looking in publicly available data, it seems comparable in those different brain regions. There's definitely some regions where it's much lower, but hippocampus and cortex, it seems similar. I would say in the mouse, where we have more experience than the monkey, there is some variability, sort of experiment to experiment, in terms of, maybe the difference between the effect size that we're getting in cortex versus hippocampus. Sometimes we see actually a little bit more in hippocampus. So I would say overall, it's comparable in those brain regions, actually.
Great. We don't unfortunately have time for questions because I was just told we have to be back here in like 15 minutes. We just had such great content. Want to thank all of our speakers, and then please seek them out for your additional questions. So thank you. Please be back here in like 15 minutes.